Chapter 9, Patterns of Inheritance, Part 2. The genetic reason behind many human traits or characters such as the color of the eye, the color of the hair, they are not very well understood. However, Mendel's laws can still be applied to the inheritance of human characters that are controlled by a single gene. That means these are monogenic characters, or this is known as monogenic inheritance. And examples of single gene characters are the presence or absence of freckles or the widow's peak hairline. The presence of a widow's peak is dominant to a straight hairline. The table on this slide shows a list of human genetic autosomal disorders that are inherited as dominant or recessive traits. These disorders are therefore they have the same inheritance pattern as was exhibited by the pea plants that Mendel studied. Most of these genetic disorders are recessive in nature. So individuals who have the recessive allele but appear to be outwardly normal are known as the carriers of the disorder. The most common example is cystic fibrosis disease, which is the lethal genetic disease. Remember the 2019 drama film 5 Feet Apart? It shows the protagonists are both patients of cystic fibrosis. So using Mendel's law, we can actually predict the fraction or the proportion of affected offspring that is likely to result from a marriage between two carriers. This slide shows how to predict offspring when both parents are carriers for albinism or any other recessive genetic disorder. Now suppose two heterozygous carriers of albinism, that means an uppercase A and a lowercase a, have a child. What is the probability that the child will display albinism? As the Punnett square in this figure shows, each child of these two carriers has a 1/4 chance of inheriting two recessive alleles. Thus, we can say that about 1/4 of the children of this couple are likely to display albinism. We can also say that a child with normal pigmentation from such a family has a 2/3 chance of being a carrier. That is, 2 out of 3 of the offspring with normal pigmentation phenotype will be an uppercase A and a lowercase a. And we can apply this same method of pedigree analysis and prediction to any genetic trait controlled by a single gene. Now, a few genetic disorders are actually caused by dominant alleles. An example is achondroplasia. This is a type of dwarfism in which the arms and the legs remain short, the head and the torso develop normally. So the homozygous dominant genotype, that means uppercase A, it is lethal. And this causes death of the embryo. That means this child will never be born because this allelic combination is lethal. So then who is the dwarf individual? It is the heterozygotes. That means an uppercase A and a lowercase a. That means the presence of a single uppercase A makes the individual dwarf because this is dominant, right? So individuals with a single copy of the defective allele have this disorder. This also means that a person with achondroplasia has a 50% chance of passing the condition on to any children. This Punnett square shows the results of a cross between a normal individual and a dwarf with achondroplasia. That means the normal individual has lowercase d, and the dwarf individual has uppercase D and lowercase d. So look at the segregation of the sperms and the eggs. So there is 50% chance that the offspring of this mating will become dwarf, will inherit the dwarfism, while the other 50% they have the chance of being normal. Now imagine, that means all of us in the world, those who do not have achondroplasia, that means more than 99.99% of the human population are homozygous for the recessive allele, which is lowercase d. So this example makes it clear that a dominant allele is not necessarily more common in a population than the corresponding recessive allele. Dominant lethal alleles are actually less common than recessive lethal alleles. One example is the allele that causes Huntington's disease, which is a disease of the nervous system. The nervous system degenerates. Now, because the allele for the Huntington's disease is dominant, any child born to a parent with the allele has a 50% chance of inheriting the allele and the disorder. What is the genetic basis of short legs in dogs? Scientists, they conducted experiments to discover the genetic basis of this short-legged phenotype in domesticated dogs. They used an automated gene chip to compare the DNA from dog breeds with short legs to that of dog breeds with long legs. And what they found is that there's a specific change in chromosome 18 that corresponded to short legs, and then the scientists were able to link this gene in dogs to a related protein associated with the most common form of dwarfism in human beings. This slide illustrates the experiment that was carried out to understand the genetic basis of chondrodysplasia in dogs. Genetic testing. In the past, the onset of symptoms was the only way to know if a person had inherited a genetic disorder. Nowadays, modern technologies offer different ways to identify about 30 treatable genetic diseases before they appear. For instance, tests can distinguish between people who have no disease-causing alleles and those who are heterozygous carriers of such disease as sickle cell anemia. Other parents may know that a dominant but late appearing disease such as Huntington's disease runs in their family, and then these people may benefit from genetic tests for dominant alleles. Information from genetic testing can inform decisions about family planning. Technologies are present nowadays for detecting genetic disorders during pregnancy, which usually requires the collection of fetal cells. And once these cells are obtained, then they can be tested or screened for chromosomal and genetic disorders. One such technique is known as amniocentesis, in which a physician uses a needle to extract about 2 teaspoons of fluid that is surrounding the developing fetus. In another type of testing called CVS or chorionic villus sampling, a physician would insert a tube into the would-be mother's cervix and remove some placental tissue. And the newest genetic screening procedures, these involve extracting small amounts of fetal cells or DNA that has been released by the fetus into the mother's bloodstream. And these new technologies are non-invasive, they are more accurate, they are safer, they can be performed earlier. Variations on Mendel's laws or going beyond Mendel's laws. Now, Mendel's two laws, that means the law of segregation and the law of independent assortment, these laws explained inheritance for sexually reproducing organisms using simple rules of probability. However, in real world, Mendel's laws, they actually stop short of explaining some patterns of genetic inheritance. Honestly speaking, for most sexually reproducing organisms, cases in which Mendel's laws can strictly account for the patterns of inheritance are actually rare. Usually we see that the observed inheritance patterns are more complex, so we have to go beyond Mendel's laws. Incomplete dominance is one of the exceptions to Mendel's laws. In incomplete dominance, one allele is partially or incompletely dominant over the other. This would result in an F1 hybrid having a phenotype intermediate between the two parental phenotypes. An example in case of human beings is familial hypercholesterolemia. This is a human trait in which this is characterized by very high levels of serum cholesterol. So the cholesterol level is very high in the blood. This happens because the receptors for that transport cholesterol are less or totally missing. In case of heterozygotes, they have about half the number of receptors to transport cholesterol, and therefore their blood cholesterol level is about twice as normal, while in case of homozygotes for this disease, there is total lack of receptors and they have about 5 times the normal amount of blood cholesterol, and this could cause a heart attack in even a child who's only 2 years of age. This figure explains the incomplete dominance in human hypercholesterolemia. This slide shows a very interesting incomplete dominance in snapdragons. So we have two varieties of snapdragons as parents, purebred snapdragons with red flowers and purebred snapdragons with white flowers. And when these are crossed, in the F1 generation, we would expect all the flowers to have the dominant phenotype. That means all the flowers should be red. But instead what we see is that the heterozygotes, because there's an uppercase R and a lowercase r, it gives the phenotype of pink colors, which is intermediate between red and white. And then in the F2 we see the reappearance of red, white flowers both, and also the pink flowers, because whenever there is a heterozygote it has pink flowers. Another exception to Mendel's laws is codominance and multiple alleles, and we see this in the ABO blood groups or blood types in case of human beings. So our blood types, you know, in human beings we have 4 different types of blood groups. We can have an A blood group, a B blood group, an AB, or an O. And these letters refer to carbohydrates or antigens which are designated as A and B, and these usually stick out from the surface of our red blood cells. So an individual with an A blood group actually has the A antigen sticking out, while the individual with B has the B antigen. An individual with AB blood group has both A and B antigens on the RBC surface, while an individual who is designated as having an O blood group actually does not have either A or B antigen. Because there are 3 alleles present for determining our blood group, there are 6 possible genotypes as shown in the figure in the next page. What are these 3 alleles? First allele is an uppercase I with an uppercase A superscript, an uppercase I with an uppercase B superscript. That means there's the A allele, the B allele, and the absence of A or B is denoted by a lowercase I. So these are the 3 alleles, and let us understand the blood grouping from the table shown in the next page. Now, if you look at this table here, the first column on the left side is the blood group type. So we have A, B, AB or O. And if you look at the second column, this shows you the various possible genotypes. Now, for a person to be A blood group, what would be the genotype for this person? It could be a person with homozygous IA, or an IA and a lowercase I, because A is dominant over the lowercase I. Similarly, for a person with blood group B, it could be an IB or an IB and lowercase I. So far it's good, but what about this AB? When an individual has both A and B alleles, these alleles are co-dominant. That means one is not dominating over the other. So they both express equally in the individual's phenotype, and this is what makes the individual an AB blood group. If you look at the column on the third column, which shows you the presence of the carbohydrates or the antigens on the surface of the red blood cell, you see for blood group A, there is this triangular carbohydrate A. For the blood group B, there is a circular one, and for the AB, there is both the triangular and the circular one. Now, for a person to be of O blood group, there is neither A and nor B. So this is the absence of A and B. You don't see any carbohydrates on the surface of the red blood cell. ABO blood groups and knowledge about these groups is very important. Matching compatible blood groups is critical for safe blood transfusions during emergencies, during surgeries. If a donor's blood cells have an antigen, that is have a carbohydrate A or B, that is not present in the recipient, that means to the recipient that particular carbohydrate or antigen is foreign, then the recipient's immune system will not accept that protein. It will consider that protein as foreign, as an enemy, and the recipient would start making antibodies against that particular protein, and those antibodies they would bind to the foreign carbohydrates, and then they would cause agglutination or clump these proteins together, and this could potentially kill the recipient because now the blood gets clotted. Okay, so there is clots in the blood because of this war going on between the recipient's antibody and the antigen that has arrived from the donor, which is foreign to the recipient. Pleiotropy or pleiotropy is defined as the phenomenon where one gene controls the action of several characters. That means a gene has more than one effect. There are so many examples, and one of the common examples is the gene that controls the color of the coat in Labrador Retriever dogs also controls the color of their nose, their lips, and the eye rims. In human beings, sickle cell anemia is a disease that is caused, that has pleiotropic effects Also, the gene that causes porphyria. Porphyria is also a disease that is also a pleiotropic gene. So in porphyria, there's deficiency of enzymes that control the formation of heme as in, required in hemoglobin. And then what happens is there is an intermediate in this biochemical reaction. It accumulates and then it is excreted with urine, or it accumulates in the tissues. And so common symptoms would be reddish teeth or photosensitivity, and this might have inspired vampire or werewolf stories. Now let us see how the sickle cell anemia gene is pleiotropic in the next slide. In sickle cell anemia, an individual who's homozygous for the sickle cell allele, so in that individual, the red blood cells, instead of being disc shaped or donut shaped, they become sickle shaped. So the sickle shaped cells, they clump together, they do not flow easily through the blood vessels, and then they accumulate and they obstruct the blood flow. So there is abnormal hemoglobin and abnormal red blood cells, and these sickle cells, they can lead to a variety of symptoms, in the downstream, and they can cause weakness, pain, organ damage, as well as paralysis. Polygenic inheritance. Polygenic inheritance means an inheritance which is controlled by more than one gene, or polygenes. And this is produced by the additive or synergistic effects of two or more genes, that means multiple genes, on a single trait, single phenotypic character. And as such, it is the logical opposite of pleiotropy. So some examples are height in people, weight, because it's not just genetics, it's also your environment and not a single gene. It's an interplay of multiple genes working there. Or susceptibility to diseases such as heart problems, diabetes, cancer, all of these show polygenic inheritance. So does it mean that here Mendel's laws are not followed? Well, each single gene trait is following Mendel's laws, but together when they come, they do not produce the same phenotypic effect as a single gene inheritance would. There is evidence that height in people is controlled by several genes that are inherited separately. In fact, scientists have identified over 180 genes that affect the human height. Now, to illustrate the principle of polygenic inheritance, this model diagram is shown here. Let us consider three hypothetical genes with tall allele for each. So let us consider A, B, and C in uppercase as the tall genes. So contributing 1 unit of tallness to the phenotype. So, and these alleles are completely dominant over their recessive alleles, which are lowercase a, b, and c. That means an individual, if he or she has all of these uppercase letters, A, B, B, C, C, all uppercase letters, that person would be extremely tall. While an individual who has all lowercase letters, all A, B, B, C, C in lowercase, this person would be really short. And a heterozygous individual with the uppercase A, lowercase a, uppercase B, lowercase b, uppercase C, lowercase c, this person would be of intermediate height or in between these two. Now, if you look at the Punnett square and with different possible combinations in which the sperm and the egg can segregate and then later fertilize or mate, so we get different kinds of individuals with different heights based on all of these permutations and combinations. So if you now plot it in a bar diagram, you find that most of the time these polygenic traits, they exhibit a bell-shaped curve as shown on the bottom right side of the figure. Epigenetics and the role of environment. Some features are determined by genes, some by environment, and some by both. But most of the phenotypic characters that we see in human beings, they are actually a result of gene-environment interaction, GEI. Now, whether human characters or traits are influenced more by genes or more by environment, this is a very hotly debated issue. This has been going on for several years. This is called nature versus nurture. Some characters are supposedly controlled more by genes, for example, your height, while some are controlled more by the environment, for example, your weight. So there are different diseases also which have more of the nature or more of nurture component. And to do the analysis for these studies, a lot of studies on twins, identical twins, non-identical twins, these have been carried out because identical twins provide us a means of studying two individuals who are genetically identical. But if, let's say, they were separated during early childhood or separated at birth and they were raised in different places, then we can study the effect of environment on these genetically identical individuals. But these are very rare cases. However, such studies have been conducted and they have greatly added to our understanding of the nature versus nurture problem. In general, the genetic influences or genetic traits are inherited by the next generation. The environmental influences, effects are not usually passed on to the next generation. You see in this figure below that as a result of environmental influences, including choices such as hairstyle or hair color, even identical twins can look very different. In recent years, scientists have begun to understand and recognize, give credit to epigenetic inheritance, which is the transmission of characters by mechanisms which is not directly changing the DNA sequence. So this mutation is not really changing the sequence of nucleotides in the DNA, but a chemical can get added to the DNA like a methyl group. Okay, so then when components of chromosomes, they get chemically modified by adding or removal of certain chemical groups, then there is effect on the DNA. This is epigenetic inheritance. So epigenetic modifications and all the changes that occur due to the epigenetic change, there is modification in the gene activity, that means modification in the protein, and this can actually be transmitted to the next generation. However, unlike the alterations to DNA sequence directly, chemical changes to the chromosomes is reversible, so it can be reversed back. Explaining Mendel's laws using the chromosomal theory of inheritance. This theory states that genes are located at specific points or positions called loci, singular would be locus, on chromosomes, and it is how the chromosomes behave during meiosis and fertilization that actually explains inheritance patterns. So it is chromosomes that undergo segregation during gamete formation and independent assortment during meiosis, and these are the ones that are responsible for Mendel's laws and Mendel's inheritance patterns. This figure correlates the results of the dihybrid cross with the movement of chromosomes through meiosis. Starting with two purebred parental plants, the diagram follows two genes on different chromosomes, one for seed shape, alleles capital R and small r, and one for seed color, alleles uppercase and lowercase y, through the F1 and F2 generation. Linkage and linked genes. Linked genes are those genes that are located very close to each other on the same chromosome, and therefore they tend to travel together during meiosis and fertilization. That means they tend to get inherited as a set. That means if gene A and B are very close, gene A and B will always be inherited together, and they therefore do not follow Mendel's law of independent assortment. So this is also going beyond the Mendel's laws. So far, the genetic inheritance patterns that we have studied, we have used genes that are located on autosomes. That means chromosome pair number 1 through 22. We haven't really studied genes that are located on sex chromosome. That means chromosome pair number 23. Human beings, we have XX or XY human chromosomes, depending on whether it's a male or a female. So now let us look at the role of sex chromosomes and the inheritance patterns exhibited by the characters they control. Many animals, including all mammals, human beings, there's a pair of sex chromosomes designated X and Y that determine an individual's sex. So individuals with one X chromosome and one Y chromosome are males, while XX individuals are females. So both human males and females, they have 44 autosomes. That means which are those chromosomes that are not sex chromosomes. This figure shows the chromosomal basis of sex determination. You can see how the male and the female have 44 autosomes each, and then there is XY or XX, and then how sperm can have an X or a Y with the 22 autosomes, while eggs are always 22 plus X. And when these meet, there's fertilization, random it's a very random process, then you can have a female or a male offspring. And there's also a micrograph on the right side which shows human X and Y chromosomes in a duplicated state. A number of human conditions, including red-green color blindness, hemophilia, muscular dystrophy, they result from sex-linked recessive alleles. Red-green color blindness is a common sex-linked disorder that is caused by a malfunction of light-sensitive cells in the eyes. A person with normal color vision can see more than 150 colors. In contrast, someone with red-green color blindness can see fewer than 25. You see the figure here, it shows a simple test for testing red-green color blindness. Mostly males are affected, but heterozygous females have some defects too. Now look at the figure, and can you see the green numerical against the reddish background? So just check it. It should be 7. If you can see 7, it's fine. If not, then probably you have some form of red-green color blindness, which is a sex-linked trait. Since the sex-linked genes are located on sex chromosomes, they exhibit unusual inheritance patterns, which is very different from autosomal inheritance. In the next slide, you will see a figure with 3 panels, A, B, and C. Panel A illustrates what happens when a colorblind male has offspring with a homozygous female who is normal for color vision. Figure B shows what happens if a female carrier mates with a male with normal color vision. And then in figure C, because the color blindness allele is recessive, a female will be colorblind only if she receives that allele on both X chromosomes, that is shown in the figure C. This figure shows the inheritance of color blindness, a sex-linked recessive trait. We use an uppercase N for the dominant normal color vision allele and a lowercase n for the recessive colorblind allele. To indicate that these alleles are on X chromosome, we show them as superscripts to the letter X. The Y chromosome does not have a gene locus for vision. Therefore, the male's phenotype results entirely from the sex-linked gene on his single X chromosome. Hemophilia is a sex-linked recessive trait which has a long history in the European royal families. Queen Victoria is supposed to be the first person who carried or in whom the mutation occurred. So what happens in hemophilia? In normal people, when we cut and there is bleeding, the blood automatically, we stop bleeding after some time because the blood automatically coagulates or clots. But in case of hemophiliacs, if there is a cut or there's an injury, the blood does not clot automatically. So they may bleed excessively when injured because hemophiliacs have inherited an abnormal allele for a factor involved in blood clotting. And the more seriously affected individuals, they may bleed to death even for relatively minor bruises or cuts. Hemophilia in the royal family of Russia. The photograph shows Queen Victoria's granddaughter Alexandra, Alexandra's husband Nicholas, who was the last Tsar of Russia, their son Alexis, and their daughters. Notice that Alexis inherited the allele for hemophilia that had been passed down for 3 generations. Evolution, connection, dog breeding, barking up the evolutionary tree. Dogs are more than man's best friend. They are also one of our longest running genetic experiments. About 15,000 years ago, people began to, in East Asia, they began to live with ancestral canines that were ancestors or predecessors of both modern wolves and dogs. So as people started living in permanent, geographically isolated places, so even their canines, the populations of canines also were separated geographically, and they eventually got more and more inbred. So a 2010 study showed that small dogs were first bred within early agricultural settlements of the Middle East around 12,000 years ago. An evolutionary tree of dog breeds. Genetic analysis has revealed the ancestral history of various dog breeds. When the dog genome was decoded, our understanding of canine evolution took a giant leap forward.